Okay, so now let's talk about ways in which DNA can be damaged and some examples of the mechanisms we have or the cells have to fix that damage. So first of all, when we're talking about DNA damage, there are different kinds of damage that can occur. A mismatch is whenever the wrong base is inserted into a growing DNA strand during replication. So this would be just usually an error by the DNA polymerase. Like if it put an adenine across from a cytosine, for example, uh, but then didn't fix itself. So this would be a mismatch that was not repaired by the intrinsic error checking mechanism of the polymerase. Um, and then an abnormal base is usually a modified version of a standard base. So a standard base that had been deaminated, for example, or otherwise modified chemically. Pyrimidine dimer is kind of a special case of an abnormal base. It's when two pyrimidines, like two T's that are next to each other, form a dimer due to UV light exposure. And then a backbone lesion is when the phosphate backbone is cut. So that can be caused by radiation or the free radicals in the cell and other things. So these are all examples of DNA damage. So it's important to note that DNA damage is not the same thing as DNA mutation. A mutation is when a damage to the DNA is not fixed and it lasts through a round of replication, and in the process of replication the presence of the damage causes the DNA polymerase to copy the DNA incorrectly. So there are different kinds. So there are different kinds of mutations depending on the way in which the polymerase makes this mistake. Substitution mutation is what it sounds like. It's when one base pair is replaced with another one. So if you had, say, an AT base pair at a certain position, and due to a mutation or a mistake in replication, you would end up with a GC base pair at that spot. And then a deletion and additions are what they sound like. So a deletion is when one or more base pairs is missing from a given sequence, and an addition is when one or more bases is inserted to a sequence. So once you have DNA damage, how does it get fixed? Uh, Mismatch repairs are kind of the most complicated, because if you have a mismatch, that means you have two bases in the two strands that are mismatched to each other, but that doesn't mean that they're both wrong. In fact, one of them is the correct base and one of them is the incorrect base. So how does the cell tell which one is which? One of the ways that can be done, or that it is done in cells, is with methylation. So when a DNA molecule is copied, the new strand eventually has methyl groups attached. So there are, in some cases, it's methylation of adenine. Sometimes it's methylation of cytosine, depends on the organism. But in any case, the methylated strand, those methylated bases, are a way of sort of labeling the parent strand, because after one round of replication for a short period of time, you have one strand that's methylated and one that's not. So this is called hemimethylated DNA. So then there are enzymes that look for mismatches, and a mismatch is detectable because if you have two bases that are across from each other, um, they can be detected by proteins that bind DNA because it causes a kink in the DNA double helix. So the repair enzymes can just fix the bases that are on the unmethylated strand, because that means it's the new strand. Now, eventually, the new strand becomes methylated, and it's not possible for those enzymes to distinguish the old strand from the new strand. So this mechanism has to work pretty quickly after replication ends. This is an example of those mismatch repair enzymes. So like I said, here you've got a mismatch right here. You have one strand that's methylated, and this is the new strand because it's not methylated. So these enzymes detect that. They can tell which is the right one and which is the wrong one, or which is the new strand and which is the old strand from the methylation. So they cleave one of the strands, the new strand, somewhere either up or downstream from the mutation, and then later a different set of enzymes comes in, detects the cut, this linear uh, break here, and then eventually cuts out a bunch of DNA, including the place where the mismatch occurred, and then replacing it all. But the point is you end up with a complete molecule which hopefully has the correct bases in place of the mismatch. Another way to repair abnormal bases, so these are bases that are modified versions of the correct bases of DNA. So, for example, uracil. Uracil is not an abnormal base, except for the fact that it never shows up in DNA. So if you have uracil in a DNA molecule, that's abnormal. So one way to fix abnormal bases is just to remove them. That's called base excision. So there are enzymes, for example, that recognize the common sorts of abnormal bases like uracil or hypoxanthine, and these enzymes called AP endonucleases. AP means apurine or apyrimidine because they can remove either purines or pyrimidines, but they just remove the base. Now they don't remove the backbone. So in other words, the strand is usually left intact, or at least there may be a cut, but there's no missing sugar phosphate backbone. So then DNA polymerase can come along and detect that and replace the missing base and some bases adjacent to it. And then DNA ligase can reconnect the two ends together. And then some kinds of specialized DNA damage, like pyrimidine dimers, have special repair mechanisms all their own. So an XI nuclease, so this is another kind of nuclease that removes, it's basically a type of endonuclease, but it removes large chunks of DNA within a strand that have large lesions. So like a pyrimidine dimer is a large lesion because it involves at least two bases and it causes a large disruption in the DNA structure. So XI nucleases can detect those and then cut on either side of them. The mechanism is the same in basically in humans and E. coli. Humans, just a bigger chunk gets cut out. DNA helicase unwinds that chunk and the whole thing gets excised and eventually broken down. And then DNA polymerase and DNA ligase can come along behind it, fill in the gap, and then reconnect the two ends. And so that's just a specialized mechanism for those kinds of errors. And then some types of DNA damage or some types of abnormal bases are so common that we've evolved specialized enzymes just for fixing those mutations. This is called direct repair. So these are just some examples here. So for pyrimidine dimers, for example, there are specialized enzymes called photolyases. Their job is just to repair pyrimidine dimers, and then alk B repairs methyladenine and methylcytosine abnormal bases, and then DNA methyltransferase specifically looks for methylguanine. So those are all abnormal bases with specific direct repair mechanisms. Now, another problem with DNA damage, apart from the creation of mutations, is that lesions can stall or stop the DNA replication mechanism. So if you have a lesion like this and you have a replication fork moving toward it, when the replication fork hits, it will stop. So the normal DNA polymerase, when it gets to a lesion like a pyrimidine dimer, for example, doesn't really have a way to proceed past it. It just kind of quits. Now, if the damage can be repaired, then it might be able to move on. But if the damage does not get repaired, then the replication process ends right here. And that's bad because basically it means the cell probably will die. And that means because the rest of the chromosome isn't going to get copied. So in order to prevent that from happening, most cells have a error-prone repair mechanism, which basically means that if there's a lesion like this, one of two things will happen. Either there will be an error-prone repair mechanism where a different DNA polymerase that's less picky basically will just put random bases here where this lesion is, and then it will just proceed on past. Or you'll enter a recombinatorial DNA repair mechanism, which we'll talk about in a second. Because in the absence, this is the same thing can happen, or even worse, can happen if you have an unrepaired break. So if one of the strands is broken and the replication fork gets here, then now the replication fork just has to quit here, because even if it could get past the lesion, there's no DNA for it to copy now. So these kinds of systems are like recombination and error-prone repair are present just for that. So next time we will talk a little bit more about DNA recombination.